Hi everyone, welcome back to my Monocle 3 video tutorial. Today I'm going to show you how to convert Seurat object into CDS object for trajectory analysis. There are three parts of my today's video tutorial. In the first part, I'm going to show you how to analyze single-cell RNA sequencing data using Seurat. I already made videos in my Seurat video tutorials. If you watched my Seurat video tutorial for data integration, you don't need to watch the first part. You can just go straight to the second part. In the second part, I will show you how to convert a Seurat object into CDS object. In the third part, we will do the trajectory analysis. So let's start from the first part. First, we need to load the packages. We need a Seurat and the tidyverse for single-cell RNA sequencing data analysis. Then we need the Seurat wrappers package to convert Seurat object into CDS object. Then for the third part, we need the package Monocle 3 to do the trajectory analysis. So the data I'm going to use for demonstration are the same data set I used in first Monocle 3 video tutorial. In the first Monocle 3 tutorial, I showed you how to download the data from MCBI website. So we have three data sets named as IPF1, 2, 3 NIN. They are the single-cell RNA sequencing data for IPF9 from 3 different patients. So let's go to R and load the data. We can load the data, use the read 10 times function to load the IPF1, then IPF2, and IPF3. So you can see in the data windows we have 3 data sets. At the moment, they are large DGC matrix data for IPF1, IPF2, and IPF3. So next we can convert them into Seurat object. Use the create Seurat object function. We will keep the cells have minimum gene expression 200, and for genes that are expressed at least in 3 cells. Now we convert IPF1. We also quantified the percentage mitochondrial DNA. Later we can use the information to perform quality control analysis. Now we can convert IPF2, get the mitochondrial information for IPF2. We do the same for the IPF3. So you can see we have 3 Seurat objects in the data windows now. Then we can merge 3 data sets into one and perform quality control together. So you can see we have an IPF Seurat object here. Then we can use the wiring plot to look at the M features and count and the percentage of mitochondrial DNA. Okay, let's zoom in. So you can see the M features and the M count has bigger variation in IPF1, IPF2, and IPF3 are very similar. So we can use, for example, the M feature below 5,000 and the M count below 20,000, and the mitochondrial DNA below 10% for the quality control. So let's subset the cells. Use M feature below 5,000 N count below 20,000, and the percentage of mitochondrial DNA below 10%. So now we can have a look at the metadata for IPF. Let's run the view function, then we can see the metadata for the IPF. You can see from the origin ident, the cells are labeled with their dataset origin. Because we are going to perform data integration for 3 datasets, so we can submit the dataset according to their origin ident. So let's submit the dataset as the IPF list by their origin ident. You can see we have a large list here with 3 elements, and we named it as the IPF list. If we click the data, you can see we have IPF1, IPF2, and IPF3 in the data list. In each dataset, you can see we have 19,299 genes, and here are the cell numbers. So now we can normalize the data one by one and also find the variable features from 3 datasets. Later we can use the variable features for data integration. So now we can select the features for data integration. We can have a look at the features. You can see here we have 2,000 genes in the features. Now we can use the features to find the integration anchors. So this will take at least 6 minutes. I'm going to have a coffee, coming back after 6 minutes. Okay, so we found all the anchors. Now we can integrate the 3 datasets. Okay, it is done. Now if we have a look at the IPF combined dataset, you can see we have 2 assays. One is RNA and the other one is integrated. The RNA has all the gene information, but the integrated only have the 2,000 gene information. They are the feature genes that we used for data integration. We need to set the default assay as integrated, then we can cluster the cells. So let's set the default assay as integrated. Then we can run the standard workflow for scale the data, run PCA, run UMAP, find the neighbors, and find the clusters. So I'm using the resolution as 0.5 to reduce the numbers for the demonstration. Okay, you can see we have 13 clusters in the dataset. Let's use the dim plot to have a look at the cell clusters. Let's zoom in. You can see we have 13 cell clusters. Cell cluster here and here are large cell clusters, and we have some small cell clusters, cluster 10, 11, and 12. So we analyzed the data before. We know cluster 10 11, 12 are cell contamination during the cell sorting by flow cytometry. Again, we can use cell markers to show cells in cluster 10, 11, and 12 are not mesenchymal cells. Let's run the feature plot for the marker genes. So collagen A2 is the marker gene for mesenchymal cells, and the PDGFA are marker gene for fibroblast, and PDGFB is the marker gene for smooth muscle cells and parasite. So let's run the feature plot for those marker genes. And we can zoom in, and you can see collagen 1A2 norm, PDGFRA, and PDGFRB. They enable the bigger cell clusters from 0 to 9. And we can use other cell markers, for example, for the epithelial cell markers, to see the small cluster of epithelial cells. Let's zoom in again. You can see markers for epithelial cells, marker cell cluster 10. Then we can run the marker genes for the endothelial cells. Let's zoom in again. You can see cell markers for endothelial cells marker the cluster 12. And then we can run the marker genes for immune cells. You can see immune cells are in the cluster 11. And we know there are small amount of mesothelial cells in the data set as well. So you can see markers for mesothelial cells enable cells in cluster 10 as well. So now we know cluster 10, 11, and 12. They are not mesenchymal cells, so we can subset the data set. Just keep cluster 0 to 9. They are mesenchymal cells. So let's subset the cells from cluster 0 to 9 and then name it as IPF mesenchymal cells. So we can use the dim plot to check if we subsetted all the cells that we wanted. We can run the dim plot. And you can see here, we only kept the cluster 0 to 9. So let's recluster the cells for the mesenchymal cells. We can run the skewed data, run PCA, run UMAP, find the neighbors, and find the cluster again. Okay, you can see with resolution 0.3 we have 8 cell clusters here. Let's see the cell cluster, use the dim plot function. And you can see we have 8 cell clusters here. You can see cluster 7 only has very few cells. So we will keep cluster 0 to 6 for trajectory analysis. Then we need to subset the cell cluster again. So let's subset the cell clusters and only keep cluster 0 to 6. Let's do the subset. We can use the dim plot to have a look at the cell cluster again. You can see now we only have 7 cell clusters from 0 to 6. Now we can use the marker genes to identify the cell clusters. We can use the feature plot function again. We know RGS5 is the marker gene for parasite, ACTA2 is the marker gene for smooth muscle cells, NAM is the marker gene for fibroblast, and ACTG2 is the marker gene for airy fibroblast. So let's run the feature plot function. And we can zoom in to have a look. You can see class 0 and 4 are parasites, class 1 are smooth muscle cells, and class 6 are ALV fibroblast. Because the cells are from IPF lung, we have a cell cluster as myofibroblast. Later we are going to run the myofibroblast markers. Cells in cluster 3 are myofibroblast. Then we know cluster 2 and 5 are alveolar fibroblast. So let's run the marker genes for the myofibroblast. We know CTHRC1, collagen 1A1, collagen 3A1, and perovskite are marker genes for myofibroblast. Let's run. And zoom in, you can see myofibroblast genes label cluster 3. So now we know cells in cluster 3 are myofibroblast. Next, we can name the cell clusters according to the expression of those marker genes. Then we can use the rename idents to name the cell cluster 0 and 4 as parasite, cluster 1 as smooth muscle cells, cluster 3 as myofibroblast, 2 and 5 are alveolar fibroblasts, and cluster 6 is the cells for ALV fibroblast. So let's name the cell clusters and then look at the cell clusters. Use the dim plot function again. We can zoom in. You can see now we named all the cell clusters for the mesenchymal cells from the IPF lung. So now we have analyzed the single cell RNA sequencing dataset. It is a Seurat object. Then we can convert this Seurat object into CDS object for trajectory analysis. Before we convert this Seurat object, we can save the object. So we just need the IPF for mesenchymal cells Seurat object for the following analysis. We can remove the rest object. Then you can see we only have one object here. So now we start the second part analysis. We need the package Seurat wrappers. In Seurat wrappers, there's a function called as cell dataset. We can convert the Seurat object into CDS object. So let's run this function to convert the Seurat object into CDS object. So you can see we have a CDS object, but you can see from here we only have 2000 genes in the CDS object. That is because we didn't change the default C for the Seurat object. Let's have a look at the default C for the Seurat object. You can see at the moment it is integrated. Because the integrated C only have 2000 genes as features for data integration. So now we can change the default C back to RNA and convert the Seurat object to CDS object again. So it is done. You can see now we have all the genes from the Seurat object, 19,299 genes. So the code data holds all the information from the metadata of Seurat object. Let's have a look at the code data. You can see we have the cells in the rows. The column names are origin ident, uncount RNA, feature RNA percentage of mitochondrial DNA. We also have the Seurat canisters here, and importantly, we named the cell canisters as parasite myofibroblast. So we can use the plot cells function in the Monocle 3 package, and we count the cells by Seurat canisters. Let's plot the cells. Because we count the cells by Seurat canisters, you can see the cell canisters are labeled by the number from 0 to 6. We can also count the cells by the ident. Let's run the plot cells function again. You can see now we have cell canisters named by the their cell types: parasite, smooth muscle cells, airy fibroblast, myofibroblast, and alveolar fibroblast. You can see now we convert the Seurat object into a CDS object. If we plot the cells, we can get exactly the same canister pattern from the CDS object as the Seurat object. If we try to do the trajectory analysis, the first function is learn the graph to see what happened from here. You can see we get an error. The error tells us no cell canisters for UMAP calculated. Please run cell canisters function. This is the function from the Monocle 3 package. So let's have a look at the CDS object. You can see from the internal code data in the list data slot, we have the UMAP information here. You can see we got all the UMAP information from the Seurat object. But in the cell canisters, we don't have any information for the cell canisters. You can see the list data for cell canisters is 0. So we need to run the function canister cells from the Monocle 3 package. Because the dataset has all the UMAP information from Seurat object, we don't need to run the UMAP anymore. We just need to run the canister cells function. Monocle 3 will canister cells according to the UMAP information from Seurat object. So let's run this function, canister cells. Okay, it is done. Let's have a look at the CDS object again. You can see now in the canisters slot, we have list data, UMAP, for the cell canisters. So we can go back to plot the cells again to see if we can get the same UMAP cell canisters as the Seurat object. Let's run the plot cells function again. You can see we have exactly the same UMAP cell canisters as the Seurat object. So we successfully converted the Seurat object into a CDS object, and we can plot the exact same UMAP cell canisters for the CDS object as the Seurat object. So now we can do the last part of analysis, trajectory analysis. So this part is very easy. The first thing we need to do is to run the learn graph function. So after this function, we can plot the cells again to see the cell canisters still looks the same as the Seurat object. Let's run. Okay, let's zoom in to have a look at the graph. You can see Monocle 3 draw the cell branches in the cell canisters. So at the moment, the color of cell canisters are labeled by their canister ident. So next step, we can order the cells according to their pseudotype. Before we order the cells, we can use the mark genes CD34 and PA16 to identify the cell canister for the progenitor cells. Then we can select the start point for the fibroblast. So let's run the Monocle cells. You can see here we got an error again. If you watched my first video of Monocle 3, when I introduced the data structure for CDS, we need to create shorter gene names for the feature data. If we don't create this shorter gene names column, then Monocle 3 cannot find the gene information. It only can find the gene ID. So let's have a look at the feature data. You can see we have 19,299 rows in the feature data. We know they are gene names, but there's no column in the feature data. We can have a look at the row names. You can see they are the gene names. So we can create a column named the gene short name and assign all the gene names in this column. Let's do this. We can have a look at the feature data again. Now you can see we have the gene names in the rows for the feature data, and also we created a column named as gene short name. The gene names are exactly the same as the row names. Now we can run the Monocle cells function again with the genes CD34, PI16. Let's run this again. You can see now we can find the gene names. Let's zoom in to see where the cell canisters for the CD34 and the PI16 positive cells. You can see these small cell canisters are the progenitor cells for non-fibroblast. So when we order the cells, we can choose here as the start point for the fibroblast. So now we can order the cells according to their pseudotype. Let's run the function order the cells. So we will open a Shiny app, and you can see here we know here is the starting point for the fibroblast canister, and for the smooth muscle cells and parasite, we can choose here as the starting point because we know the progenitor cells are in the canister of smooth muscle cells. So let's choose two starting point, then click the done button. Now we can go back to R and see the pseudotype. We just need to plot the cells colored by the pseudotype. Let's run. We can zoom in to have a look at the pseudotype. So you can see for the fibroblast, we label the next point as the start point. So the progenitor cells generate the alveolar fibroblast, then they became myofibroblast in fibrotic lung. And for the smooth muscle cells and the parasites canisters, you can see the cells starts from here, and all the fibrotic parasites could be derived from the progenitors of smooth muscle cells. So for the trajectory analysis using Monocle 3, you have to label the start point. If you label the start point from different site, the field time map will look different. I think Monocle 3 is very good for trajectory analysis if you know the start point. For example, if you differentiate organ-specific cells from stem cells, or you know the progenitor cells for tissue development. But for identify the myofibroblast origin from the fibrotic lung, I think the RNA velocity analysis is a better method than Monocle 3. Okay, I'm going to stop here. In summary, I showed you how to analyze single RNA sequencing data in Seurat, then convert Seurat object into CDS object and do the trajectory analysis. So I hope my video tutorial could help your data analysis. Please subscribe my channel and share my channel with your friends. Thank you and see you in my next video tutorial.